The overall goal of this procedure is to determine the structure of a metallochaperone protein mimetic peptide complexed with copper. This is accomplished by first preparing the sample in an oxygen-free environment. The second step is to acquire the nuclear magnetic resonance, or NMR, spectroscopy data showing interactions among hydrogen atoms. Next, the spatial interactions are mapped onto a linear peptide template. The final step is to find a representative low-energy structure that fits the data. Ultimately, NMR-derived structures are used to determine the mode of binding and to perform structural analysis on the copper-bound complex.
The main advantage of this technique over existing techniques such as X-ray crystallography is that you can use it to look at weakly binding complexes and also at molecules and complexes that don't crystallize.
Though this method can provide insight into copper-binding proteins, it can also be applied to other systems, such as other metallochaperones, to study how proteins enable safe delivery of essential yet potentially toxic metal ions in the cellular environment.
To begin, prepare samples in an oxygen-free environment to prevent the copper from oxidizing. To prepare the APO sample, dissolve approximately 1 to 2 milligrams of the peptide in 450 to 500 microliters of deuterated NMR-grade solvent. For the copper-reacted sample,